All right, so before we get started, just a couple of disclaimers. Please note that the information presented during this session is not medical advice, nor is it intended to be a substitute for medical advice, diagnosis, or treatment. Always seek the advice of your physician or another qualified health provider with questions concerning a medical condition. The Immune Deficiency Foundation education events offer a wide array of educational presentations, including presentations developed by healthcare and life management professionals invited to serve as presenters. The views and opinions expressed by guest speakers do not necessarily reflect the views and opinions of the Foundation. With that, I'm going to go ahead and briefly introduce our speakers. You can find their full bios in the conference app. So I'm starting actually in the middle for the bios, but Dr. Xiao Peng is a physician scientist and rising star in the field of primary immunodeficiency disorders at Johns Hopkins University in Baltimore, Maryland. Her lab focuses on unraveling the mechanisms of various primary immunodeficiencies with a specific interest in studying the development, function, and regulation of T cells and B cells. Do I have the wrong bio for you?
It is very possible.
Okay, I do know the Hopkins part is right.
Yes.
Okay, well, sorry about that.
Summarize?
Sure.
Hey guys, good morning. Who needs more coffee? All right, so I'm Xiao, nice to meet you all. I'm the director of the Genetics of Blood and Immunity Service at Hopkins, and so what we really try to do is provide sort of wraparound diagnostic and management for inpatients and outpatients who have genetically driven blood and immune disorders of all ages. Nice to meet you.
All right, thank you. Hopefully the next bio is a little bit more accurate, but you'll let me know, I guess. Dr. Magdalena Wakiewicz is a distinguished scientist specializing in primary immunodeficiency disorders at the National Institutes of Health in Bethesda, Maryland. She leads the NIAID centralized sequencing program at NIH, focused on understanding the development, function, and regulation of the immune system. All right, woo! And on the end here, we've got Dr. Sung-Young Pai is a physician scientist, tenured senior investigator, and chief of the Immune Deficiency Cellular Therapy Program at the National Cancer Institute, also at the National Institutes of Health. Her research focuses on inborn errors of immunity and their treatment with allogeneic hematopoietic stem cell transplant and autologous gene therapy using viral vectors. All right, woo! 2 for 3. All right. Thank you all for being here. This is a little bit of a less formal session. So instead of presentations, they're each just going to be talking about some of the studies that they are doing. And then we'll, like I said, do questions at the end. All right. Thank you.
Speak a little louder.
I'm just going to take this off.
Oh, it's not working at all.
No, I don't think so. Good morning, everyone, and thank you for inviting us to come to this wonderful meeting and meeting the families that we hope to serve. My name is Magda Walkiewicz, and I am trained clinical molecular geneticist, and what that means is that my main interest really is in the genetics and genomics of immune deficiencies. I have the pleasure and privilege to work at the NIH, specifically at NIAID, and my main interest really is to collect DNA from our patients and their families, then sequence the DNA and look for molecular defects that underlie disease of our patients or our referrals. Our program, or my study, started 7 years ago when I came to lead this centralized sequencing program. We initially were really just doing whole exome sequencing we offered whole exome sequencing to every patient that walked into the door of NIAID. We then moved from exome sequencing to short-read genome sequencing. This is much more comprehensive study. It allows us to look at the entire genome of any human that agrees to our protocol. And then we really are interested in many layers of that data. First and foremost, we really look at the Mendelian predisposition to immune deficiencies, right? So one mutation that causes a disease, we welcome anyone to come to NIH. If you already have a molecular diagnosis, you still are welcome to participate in our study. We are interested in looking at that mutation from our lens, and then we are also interested in looking at other variants that might be present in the genome that might be changing or might be affecting how that disease progresses in you or your child or family members. We know that there are differences. We know that not everyone presents the same way. And we suspect that in addition to environmental factors and so forth, there are also genetic factors that affect how that disease progresses in a family or in an individual. In addition to the Mendelian diseases, there is also a lot of other changes that we are interested in looking at. For families where we are not finding a mutation that may be responsible for their phenotype, we hypothesize that it may be a lot of small impact variants together are driving some of the phenotypes that we see at NIAID. This is a relatively new study, and it really requires, or relatively new approach to PID, I would say, and it really requires large cohorts of patients because we really need to look at a lot of people to help us understand what's typical and where is that threshold that pushes you over to developing a disease. So one of the studies that we are really interested at looking at is aspect of the research that we do is also collecting families without a Mendelian disease and just keeping that data on our radar and looking at these families that look very similar and combining that data together and trying to understand whether there are these small defects that together cause a disease. In addition to small variants, we are also interested in looking at large variants, so deletions and duplications. And it's actually interesting because that has not really been a main focus of PID for a while. We did a study when we came to, when we had about 2,000 patients in our cohort, and we actually found that 10% of the patients that we've studied at NIH did not have a small variant. They actually had a large deletion or a large duplication. So, you know, you are teaching us how to look at the data, and you're helping us to understand how the genomic architecture of human beings affects the immune system. And this has been super valuable in understanding and then developing follow-up hypotheses of the research that we are doing. I think one of the really wonderful things about the NIH is that, you know, we do the genomic part and then we hand it off to the many investigators that collaborate with us. And then these findings that these genomic or genetic findings that we have, then they can really guide the treatment for your families or your children or yourselves. And this is just a very unique environment that allows us to not only discover new diseases, but also allows us to tailor the treatment for these diseases. I think there is a lot of, bone marrow transplant is one of the avenues, and it's always so important to know what the molecular defect in a family undergoing bone marrow transplant is, because some of the family members may also carry the same variant, but might not be presenting the disease. And this really is a very, very useful information in deciding who the donors are for families. discovering a new disease, understanding the biology of that disease really can inform treatment and tailor treatment so that you can really target a pathway that's responsible for certain disease and so forth. And the NIH really provides a very unique environment where we might be finding a new gene or a new mutation, and then we can just hand it off to the scientists in our institute who can take it from that particular information all the way to treatment. And I think there's just so many examples of that, and it just creates such a unique environment for patients and their families. I think collaborating with people in our field, sometimes we find something, and there are also a lot of investigators around the country and internationally who study similar diseases, and sharing that information really just allows us to move the field forward. We meet at conferences, and we are, I would hope, we are extremely collaborative, and we are always interested in exchanging that information. And I think in many ways, the field of genetics and genomics in PID has moved so quickly. We have so many new discoveries that I think are just absolutely breathtaking in how they directed treatment for so many of our participants. But I also think that in the next few years, with the technology moving so quickly, we're going to be discovering even more and more gene defects and other types of variants that we were not even able to look at until this time because we just didn't have the technology to look at. I also just want to point you to the outstanding research that is going on at the NIH and outside. For example, the Broad Institute has been on the forefront of sequencing healthy individuals because that data is equally important. It helps us to compare the healthy individuals to the non-healthy individuals and draw conclusions about genomics and the genetic architecture of PID and so forth. So I am super excited to be at the NIH and leading this study. I think we have so much to discover. I think our research is just generally, the way I like to think about it, it's really where we just start unraveling some of the mechanisms of the biology of PID, and then how the biology and pathophysiology leads to the disease. And it's just been a wonderful opportunity. And for anyone who ever came to the NIH and donated their time and their sample to us, I'd just like to thank you. And for everyone else, if you are interested in coming, you're welcome, and we would love to have you on our study.
Hey guys, so I think it's great that Magda started off because then she said a lot of things I wouldn't need to, but I actually wanted to step back and talk about some more, I guess you could say, home truths about where we are in the field right now. So I think we're all really, really lucky to be living in this time. And I do want to take a moment to sort of marvel at the fact that, you know, it was only 11 years ago or so that we were able to publish the first draft of the human genome, which took 13 years, 20-something institutions, and thousands of 3730 Sanger sequencing machines that are larger than the table here. Now, when you go to a doctor, you get a cheek swab, and you can get some of your results back within, you know, sometimes 7, sometimes 3 days, right? So a lot of times, you know, we forget that we're very much still at the beginning of this molecular diagnostic journey, and it's something that all of our clinical providers are having to come to terms with, and certainly all of our patients, because I often get patients saying to me, well, I was already given a diagnosis of lupus, of ALPS, of CVID. Why are you now telling me I have another diagnosis, NF-kappa B deficiency, a TACI mutation, CGD? So, you know, this is this whole world we're facing now where you're going to have a lot of labels, right? You not only have the clinical labels that we've been able to give people sort of in the same way we've been able to assign paintings to famous artists, right? From what we can see, all the things we were able to measure using clinical tools. But now you have the ability to sort of look more on a molecular and cellular level and get at a diagnosis that's molecular, that can sort of explain what's going on in all of our cells. So it's not like it's conflicting information. It is actually all different pieces of information that can all be used and should all be used together in a synthesis. But sometimes this is really quite hard, actually, for a lot of clinicians to even understand. The other thing is, I come from a background, actually, of yeast genetics. So I'm used to working with the single-celled organisms that make your beer and bread and wine. And that always reminds me that immunity is not limited to the immune system or to immune cells, right? So there's immunity in every living cell, whether it's yeast or plants or from human cells, whether it's our neurons or our skin or our gut. I think you've heard a lot of examples of this from a lot of the other speakers who've spoken before me. So the interesting thing is we have a lot of amazing assays being developed for our immune cells and how to interrogate the processes that are driving immune disease in those cells. And that's incredibly valuable for geneticists to learn about and to use together with our genomics technologies. And it's something I'm very passionate about, sort of bridging that gap between all of these different kinds of tools that we can use. Sequencing is just another tool. But there's also a lot of hidden information that we can't measure because no one is going to stick your brain to do an assay, hopefully. No one is going to, do some of these tests. No one has the ability currently to do some of these clinical grade tests in your non-immune cells, even if those might be the culprits of your immune disease. And so that's kind of where a lot of the burden on the geneticist comes in, right? Because we can sort of interrogate your cells in a much less invasive way and try to get at some of those more difficult to diagnose immune diseases where the problem is not just in your immune cells, but really in many different cell types. And that certainly impacts the treatments we think about. Another thing I wanted to talk about is one of the exciting things about working in the blood and immune realm as a geneticist is that it's a really unique genetic landscape, which seems to drive a lot of other geneticists nuts who are not like Magda and me. So I think it really selects for a personality that actually likes complexity and likes to be surprised. A lot of the rules that we follow and a lot of the rules that geneticists have sort of designed for not only discovering disease-causing variation, but also classifying them, don't really work that well for our genes. And so we kind of have to pave our own paths in a lot of ways, which is why we, spend a lot of time talking to each other and helping each other sort of navigate a lot of the interesting paradigms that exist. Magda talked about one of them, which is this idea that we may have oligogenic causes of immune disease that are, combinations. You'll probably read in one of your patient newsletters, mention of structural variants and somatic variation. These are things we lose sleep over every night for our patients that someone else may not as much have to. And we also have to come up with tools to address some of those complexities. The really other important thing I want to talk about is sort of, right these days, we do rely a lot on omics technologies, whether it's DNA-based genomics or RNA-seq-based transcriptomics or any other omics that you might probably have heard of. But really, at the end of the day, nothing quite substitutes for a really, really good clinical history. We can't find anything without your help. And I will tell you that without parents of some of my patients having noticed certain things about what triggered the episodes that their children had, we would have arrived at a very different conclusion about the origin of their symptoms. So it's incredibly important to share your suspicions, share your hypotheses, share your observations with your clinicians. It's equally important that they listen to them. The other really both annoying and cool thing about genetically driven immune disease is that oftentimes two people in the same family with the same exact mutation can look very, very different. That's usually the rule, not the exception. Whereas two people with totally different mutations and totally different genes may look very similar, both clinically and immunophenotypically. So this is this presents kind of a challenge for those of us who are trying to do genetic analysis on the basis of feature matching, right? And it means that as a field, we need to move more towards what I call pathobiology matching, which is not listing the genes that have that particular symptom, but really thinking, well, what could cause this? What kinds of processes going wrong in the cell can lead to these possibilities, and trying to bark up those trees rather than looking at the leaves or the stems and simply trying to match those. So I think that's a really, really important thing, both for diagnosis and also for management, because a lot of times, you know, we may not get an answer from our fancy sequencing for quite a while for our patients, but we still need to keep them alive. We still need to improve their quality of life, right? And so we can actually use those immunophenotypic pieces of information, those similarities, to our advantage, and actually use some of that information to say, okay, well, what this person's immune cells are doing actually looks like this other patient I have who has a known diagnosis. Can we try to extrapolate from that lesson and treat them in the same way? And we've been able to do that successfully while we continue to pursue molecular diagnosis for our patients. That's one of the things I really like about this field, is that just because you don't get a molecular diagnosis up front doesn't mean there's not things that your doctors can still do for you. So having said all of that, you know, what I do for a living is really very, very simple, and really is kind of the mission of, I would say, almost all of the geneticists who work in the world, which is to bring the appropriate diagnosis and treatment to their patients. There's nothing fancy or original about that. I think the utility of my sitting here talking to you in comparison to Magda's story is that, you know, we at Hopkins, we do have to work with insurance, and we have to work with a lot of the constraints that, for example, some large institutions and certainly federally funded institutions don't struggle with. So a lot of my daily grind is arguing with insurance, right? And you guys all, I'm sure, know what that feels like. So, you know, it's not enough just to develop third generation long read or optical genome mapping or all of these, you know, strategies that are going to address how to detect those difficult to detect lesions, whether it's, you know, big deletions we can't pick up, deletions in very strange regions, you know, changes in repetitive regions, things like that. I think we all understand where our missing heritability is, but we also do need to be able to address the practical issue of how do we develop these as clinical tests. How do we make sure the FDA doesn't stranglehold us from trying to get these into patients' hands? And how do we get insurance to actually pay for this? And some of that is sort of, you know, a lot of ongoing discussion, but a lot of it depends on having participation from our patients in these pilot studies so that we can show that there is true impact, both from the genomic and the functional and any other types of assays that we really use to try to help our patients. So I think at the end of the day, I just want to say thank you guys so much for continuing to help us fight this battle. And it's a privilege to take care of you all.
So hello, can you hear me? Does this work? Okay. I'm Sung-Yun Pai. I'm a pediatric hematologist-oncologist, and I first became interested in immunology when I was a medical student. I worked in a laboratory that studied how agents like cyclosporin and tacrolimus and rapamycin work in the cell. And then when I was a first-year fellow in Hem-Onc, my first transplant patient was a little boy with Witt-Scott-Aldrich syndrome. And that led eventually into making transplantation and gene therapy for inborn errors of immunity my specialty. And when I was training, the approach that we took was pretty much the same for every disease. And over time, I've become convinced and committed to the idea that we should be tailoring the treatment to the biology of the individual disease, with the ultimate, of course, being, instead of allogeneic transplantation, gene therapy. And you know, this is important because the approach should be different, and the outcome of how well the patient's disease is corrected when there's mixed chimerism, a mixture of donor and patient cells, is related to the biology of the disease. So I was faculty at Boston Children's Hospital for 19 years, and I had a laboratory and a clinical research program in this area. In November of 2020, I decided I was tired of dealing with insurance and that I had an opportunity to join and also lead a group of investigators who are interested in the same thing. So that's why I moved to the NIH in November of 2020 during the pandemic. It was a little bit weird. And I do miss Boston a great deal, but the NIH is a wonderful place to work, as has already been mentioned. In terms of the trials that I am either leading or involved with, there are two types of trials. One are trials that I started when I was back in Boston that are targeted to infants, and then the other are trials at the NIH that are for children and also adults. So when I was in Boston, I started two trials for SCID. One is the Conditioning SCID Infants Diagnosed Earlier Seaside trial. This is a trial that continues to enroll at 50 institutions in North America. And we are trying to test the question of when you give conditioning to a SCID patient of particular genetic types, how much do you need to give in order for both T cell and B cell function to occur? And this is based on work that I did with the Primary Immune Deficiency Treatment Consortium, where we figured out that certain types of SCID really do need conditioning for full immune reconstitution. But we would like to use less if we can get away with it, and we think it might be different for different types of SCID. So that's the Seaside study. And the other is an X-linked SCID gene therapy study, which is ongoing using a lentiviral vector and using a very low dose of busulfan in order, again, to reconstitute both T and B cell immunity. Those trials enroll infants, and at the NIH, we do not enroll children under 3 years of age, specifically also in transplant or gene therapy trials. So when I moved to the NIH, instead I've become involved in trials that are targeting older children and adults, and especially the treatment of adults with allogeneic transplant and gene therapy is quite unique to the NIH. I'm not sure there is any institution in the United States that has as much accumulated experience in transplanting adults with immunodeficiency. I'm very fortunate, I'm at the National Cancer Institute, to be part of a group with NIAID investigators. When I first got to NIH, well, starting about a year before I got to NIH, there had been siloed research programs in clinical transplantation, and so there was like 5 different clinical teams. And this was a little bit crazy, and also the pediatric patients were kind of scattered through the adult teams and were not necessarily cared for by pediatricians. So before I got there, and then I sort of completed the process, we've now consolidated the services into an adult transplant service for non-malignant disease and genetic disease, and an adult service for leukemia and other cancer, and then a dedicated pediatric service. So now what we try to do is when there are patients who are interested in enrolling in one of our trials, the NIAID and the NCI investigators have a joint conference and a joint evaluation system so that we can serve as kind of a clearinghouse and try to match patients to the most appropriate protocol. The protocols are 2 different kinds. There are some that are specific to a given disease. So we have a couple transplant protocols for GATA2 deficiency, one for DOC8 deficiency, several for CGD, including gene therapy. And then we have some protocols that enroll many different types of immunodeficiency. And while a lot of different types might be eligible, we try to focus on the ones where the NIAID research interests and the NCI research interests are aligned so that we can accumulate patients with a given type of PI and really make some progress in how the treatment should be best done for that disease. So I think I'll stop there.
right, thank you all. Before we get to the audience questions, I do have a couple of questions myself, and any one of you are free to answer these. What's the best way for everybody out in the audience to stay up to date with these studies and what you're finding?
Clinicaltrials.gov and Googling.
Yes, Google it. So yeah, just to expand on that a little bit, if you are unaware, clinicaltrials.gov lists every clinical trial with human subjects that is happening, right? So that is basically your Google for clinical trials.
Yeah, and then I think from the diagnostic side, because we are not the same kind of study, we are usually not in clinical trials. I think either looking on PubMed to see, what people have been publishing, and also just visiting the websites of the researchers is usually helpful. And I've noticed that a lot of our partner sequencing companies actually are very proactive about posting on sites like LinkedIn or Instagram or X, or terrible at social media. But yeah, those actually, they usually post a lot of these, especially as new chemistries and new technologies come online. You'll see a lot of that information. So Google is often a good option.
All right. And are there other studies that you all are excited about? I know that you were saying 1 million that perhaps you're not directly involved with, but are watching closely.
I wouldn't point to specific trials per se, but I think it's very exciting in the treatment field to be seeing base editing, prime editing, newer ways of genetic manipulation of stem cells to treat PI. The other is non-toxic conditioning. So everything that we do in transplanted gene therapy right now, in order to make space for the new stem cells, either from the donor or the ones that have been genetically manipulated, requires chemotherapy. And some of the limitations on who we can take to transplant or gene therapy is based on the patient's ability to tolerate that chemotherapy. And in children especially, this leads to long-term side effects that people have to then live with, through adulthood and older adulthood. So I think the non-toxic conditioning combined with the newer gene editing could really change the entire landscape of these treatments.
Yeah, I definitely second that, especially, I take care of a good amount of patients who have genome maintenance disorders where their specific genetic defect basically prevents them from getting a lot of the standard chemotherapy regimens. So the non-toxic conditioning thing is something really, really exciting for us. I also want to point to this idea that is growing in our field of doing basket trials, where instead of having, let's say, people who have a specific group of mutations in one gene, they take people who have shared biology from having different defects in genes in that pathway, which gives you so much more power, especially when you're dealing with rare disease. So there's a really exciting trial going on right now with JAK-STAT gain-of-function disorders, and I'm sure we'll see more going on in the future, certainly for PI3 kinase gain-of-signaling, for some of the NF-kappa B disorders, you name it. So really going back to that idea of sort of grouping patients more by pathobiology than specific mutations or specific symptoms. From a diagnostic side, I think we're learning a lot of lessons from each other and also from our European colleagues where, certainly socialized medicine has made it a little bit easier for them to gain traction with a lot of these newer sequencing technologies sooner. But one of the most exciting things in the field has been to be able to see some of these like larger national or even pan-European cohorts begin to publish the first experiences and results that they have had with what we call third generation or long read sequencing.
Yeah, I am actually super excited about the new I'm very excited about the new technologies that are emerging. I think, for you, The cost of sequencing is probably going to go, it's going to just decline as it has been. I think some ability to study the places of the genome that we were not able to study before, that is really emerging at a pretty fast pace. And then just outside of genomics, all the other omics, I think it's equally, I think it's equally quickly becoming much cheaper and much quicker and much more accessible to a lot of researchers. So I really think the next 20 years are just going to be a gold mine for research and for understanding PID.
One thing I will add is actually, a lot of the cost limitations now aren't necessarily in the actual generation of the whatever omics technology. It's actually in the cost of storing and processing all of that data. So that's certainly one of the current bottlenecks. And having actual, people and programs be able to analyze those appropriately.
All right, thank you so much. Before we open it up for questions from you all, there's just, here we go, sorry. A couple of resources that I wanted to point you to. The QR codes up here go directly to these programs, so definitely get those on your phone. Ask IDF, so this is our online basically answering service. You send us your question about PI. It can be insurance related. It can be, I need to find a clinician in my area, those types of things. And we will either send you an answer, a resource, or point you in the right direction. I will say, just as with the conference sessions, we can't offer medical advice through Ask IDF, but we can definitely get you pointed in the right direction. If you need peer support, we have these great Get Connected groups. Some of them are diagnosis-based, some of them are geographically based, even though they're online. So you can meet others and just get to know some of the people in your area that have PI. The last thing I want to say is please, please, please complete your session survey through the conference app. This is the only way we know if the content we're providing you in these sessions is actually meeting your needs. So we have to hear from you, otherwise we're kind of flying blind. And with that, we're going to get to questions. My lovely colleague in the back has the microphone. A couple of ground rules. Please keep the questions relevant to today's topic and do not disclose any personal health information. We also ask that you not name specific brands of medication, specific providers, or specific healthcare institutions. And with that, Colleen's going to bring around the microphone.
First question, I'm going to give you guys a prompt here to give some people some instruction. When they get to clinicaltrials.gov, they feel like they're at the IRS site. So how do they progress? What's the difference between an investigational and a treatment trial? And then how are they expected to inquire?
So those of you who have, let's say, a known diagnosis, you can enter the diagnosis, or you can enter a type of therapy or a drug name, whatever it is in these search fields. Once you get to your list of search results, it will say what kind of trial it is, whether it's a natural history study, whether it's an interventional study, whether it's a treatment study, or whether it's more of a research understanding the biology study. And then every study has a contact information, who is the leader of the study, and then who to contact for more information.
I have a question on some of the studies that you are in now, or future studies. I did not hear, maybe I missed it, anything on natural killer cells to be reconstituted or for an adult patient.
By the way, I was so moved by the film yesterday, so thank you so much for everything. I I didn't say anything about NK cells. I'm assuming you're talking about NK cell reconstitution and function after an unconditioned transplant for X-linked SCID. And this happens to be a bit of an interest of mine. I don't have specific studies per se, but there are researchers at NIAID who are working with me to try to understand what is the role of NK cells and whether NK cells not reconstituting, what kind of impact it has, and which problems that some patients with SCID after transplant face can be corrected and how we can correct them. So I'm happy to speak to you more about it in the future.
How would you recommend someone with PI and multiple genetic variants get involved or get in touch with the NIH to be a participant in a study?
You can contact me directly, and I'll point you to the right people. I just want to let you know that to participate in my study, there's a small requirement that you have to have a you need to be enrolled on a protocol by a primary researcher. So for example, if you have a variant in PIK3CD that we don't know what it means, you can get involved with Dr. Uzel as the primary researcher, and then you co-enroll on our protocol and we'll study your genomic data.
Hello, I have a question. I actually have a two-part question, if that's okay. The first one. So for somebody who acquired hypogammaglobulinemia from treatments, chemotherapy drugs that I won't name, that did lasting damage to the bone marrow. There doesn't seem to be a lot of studies on PubMed specifically about bone marrow transplants or other treatment options for people who weren't born with a genetic mutation that led to hypogamma. It's mostly people who were born with it or children. I guess my question for this first part is, are there any ongoing studies or new studies for people who have acquired this damage from these drugs, or any other treatments to reverse the DNA damage that's incurred that has messed up humoral immunity?
So I think, it's interesting how you framed it, people who were born with hypogamma versus not. Just because a person wasn't born with hypogammaglobulinemia, just because a person needed a trigger of some kind to develop hypogammaglobulinemia doesn't mean they don't necessarily have an underlying genetically driven predisposition. And there's been many observations now with certain types of drugs that are used for people that I will not name that induce, persistent chronic hypogammaglobulinemia and B-cell dysfunction that some people seem to recover very well from, but a third or so of patients never recover from. So there is actually a lot of interest in the field with respect to that population who never recovers B-cell function. I can't tell you specific studies, but I can tell you that has been an ongoing source of interest because we do suspect that at least a subset of those patients probably do have genetic factors that have influenced that outcome.
I do know one researcher who's particularly interested in the lack of recovery of B-cell function after certain treatments, so come find me later.
But, I mean, if you came to me and just said that, we would be happy to genetically test you. That in and of itself would be a cause for a genetic evaluation from any geneticist who does immune disease.
And I fully agree that, both could be going on. There could be an inborn error. It could be that it was purely treatment related. It could be a combination, because sometimes the inborn error that is predisposing to a cancer that then gets treated, so both can occur.
There's a lot of situations where the environmental impact is actually the thing that uncovers the genetic influence, and so we always have to consider that. It's rarely just one particular contribution, and it's rarely always obvious.
Thank you for what you're all doing. My basic question is, sometimes it takes too long for the results of these clinical trials to help our patients. What are your ideas, or what are the roadblocks that need to be removed so we can bring those treatment to our patients sooner so they suffer less?
I think there are two different parts of that question. One is how long does it take for the results to be disseminated? But I think what you're really asking is, especially in the field of, let's say, gene therapy, what is the barrier once the gene therapy has been shown in an early phase trial to be effective to getting it to be a licensed treatment? And this is this is one of the things that keeps me up at night because, as you know, in the PI field, there was a period of time where we thought, oh, all we have to do as the academic researchers is to conduct these trials, show that it works, and then a company will magically take it and bring it to people. And that really hasn't been happening in the way that we wish. I think that we need a new model. We need, this reliance on industry is probably not going to be the main solution, and we need a new model. This is something that I've been trying to discuss within the gene therapy community so that we can find a different way to bring the patients the therapy, but there's some hurdles. But the way that the IDF community can help is by doing advocacy. As a federal employee, I cannot do any lobbying. And so I'm relying on you all to bring your stories to Capitol Hill, to explain that this barrier must be overcome, and that the government needs to act.
Over here.
If you've already gone through genetic testing for an NF-kappa B mutation, and there's been so many changes that have come up over the last, 5-7 years, and you went through testing 10 years ago, is there a benefit to redoing testing now so that you can see if you fall into some of these rare disease categories? And then second part, when a doctor is in a research study at NIH and chooses to leave and you can't find information on the study they were originally doing, what are the next steps to kind of trying to get back into that pathway to get answers and go? Timing didn't work out the first time.
I'm going to add to that first question. I think a lot of people have that question. How often should we go through genetic testing?
So this is how I would answer the question. If you have a molecular diagnosis that your doctor, the physician that you're seeing agrees with the test results, I would probably stop there. If you have a variant of uncertain significance on that report, I would keep going. And there are a panel, so a lot of you probably had panel sequencing, right? So it's a limited number of genes that were looked at certain time. 10 years ago, the coverage, so how well we were sequencing each gene on that panel, is not as good as it is today. In addition, I personally, and it's easy for me to say that because I work at the NIH and I don't deal with insurance. I personally would not do a panel testing at this point in time anymore. I would do either exome or, if possible, genome sequencing, because that data is really good today. It can sit for the next 10 years or so, and it can be relooked if we don't have a diagnosis today. So the second point to that question is, our knowledge over the last 10 years has gone from about 200 genes in PID to about 500 genes that we know of. And I can assure you there are a lot of more in the pipeline to be published and worked up. So if you have exome or genome sequencing in the last 5 years, I would ask the lab that did the testing to take a look at that data today, because it is possible that there was a gene defect that at the time that someone looked at that data 5 years ago was not published. We didn't know it causes disease, but someone discovered it, published it, and now it's a known disease gene association, and relooking that data today might lead to a diagnosis.
add a little bit to that. So Magda mentioned two main groups of confounding or sort of issues, right? So one is the actual clinical validity and utility of your test and where that is. And another is the technical analysis component. So with respect to testing, forget 10 years ago, there's tests that my patients have gotten last month that are not the same as the one they've gotten this month. A good example, for example, is there's a genetic testing company I won't mention, but they recently updated their exon capture kit. So if you happen to have a mutation in Icarus, the IKZF1 gene, then you had a 10% detection ability or, let's say coverage in September of 2023. After the exon capture kit got switched, you went up to 100% detection. So these are nuances your geneticist needs to be aware of, not you. It's not your job to memorize these things. The second thing is analysis also is constantly improving. So just because you have a variant and it was picked up on sequencing doesn't always mean that the lab is going to find it. There are regions that labs blacklist out from their analysis. There are, analysis is still largely a manual or an AI-driven operation, but either way, if they don't understand the paradigms behind those genes, they may miss them. So you also, certainly at the NIH, there's often reanalysis being conducted for exactly that reason, where people go and they physically take a look, with domain experts who understand the nuances of the genetics behind those variants. And of course, we are constantly discovering new genes associated with immune disease, and clinical grade genetic testing is only allowed to report known bona fide gene-disease relationships, right? So if it's not been published, it's oftentimes disregarded, even when it has been published. I will tell you the genetics field usually waits for multiple papers to publish something, whereas in our field, if there's one really good example, we take it, right? So there's a little bit of difference there as well. So all of these nuances really are sort of the responsibility of the person who is doing your molecular diagnosis to sort of keep up with. Actually, a lot of times if you have VUSs, there may be other ways to get at that VUS, whether it's through clinical grade immune studies or research grade immune studies. And so another way to approach it is to try to prove or disprove that some of those VUSs are potentially relevant to you, in parallel or series with additional genetic testing. I will, though, say, do not discount old genetic testing. I've diagnosed a patient from something that, from a region of homozygosity that was found on a test 15 years ago. It was still valid. So again, having clinical suspicion is also really, really important, and making use of all the data available, but certainly making sure that you're aware of the current limitations and specific detection capacities of specific tests, because I think oftentimes we assume all gene panels are created the same, or all exomes or all genomes are created the same, and that's actually not true. So reading the fine print is really important.
All right, so we have run out of time. I know there are a lot of questions still out there. So again, ask IDF. We're happy to take your questions and point you in the right direction. I want to thank all of you so much for sharing your expertise.